went to the FDA. This was the initial trial design that was approved by the FDA for all 3 of the different clinical trials that we have approved in the US and for the Stargardt's trial in the UK. It is a 12-patient trial design, cohorts of 3 patients each, ascending dosage format. When we initially started the study, we had been approved to treat patients who were very end stage in each of these diseases. 2,800 was the best vision we could treat. From 2,800, it's counting fingers and hand motion to give you a sense of how far down the spectrum of visual loss we were at. Based on the data from the first cohort of patients, we went back to the FDA and to the MHRA, and they allowed us to modify the inclusion criteria to move this up to patients as good as 2,400. Halfway through the trial, based on the very good safety data that we had for at that point, both sets of patients in the U.S. trials as well as those in the U.K., and the fact that we were seeing improvements in visual acuity, we went back to the FDA, and this time we asked them to allow us to continue with the 100,000 cells, which we'd already crossed the safety profile for, but now to do it in a special cohort of patients with vision as good as 2,100. So the goal of this was, so we have in the US, in both the Stargardt's and the dry AMD trial, we have the ability to test 4 patients in each of those studies at this better visual acuity. The goal was to head towards patients who represent intermediate AMD rather than very late stage AMD or very late stage Stargardt's disease, and to start to kind of tease out what the potency or efficacy of these cells might be as we think about a phase 2 study. So that's where we are. I'll tell you in a minute where we are in the trials altogether. We, as a little company, I think I'm very proud of who we've been able to attract into these studies. We've managed to pick up 4 of the top 5 eye hospitals in the U.S. as clinical trial sites. The only one that's missing from that list is Wilmer Eye at Johns Hopkins, and they have a member of their surgical team who's actually part of our data safety management board. So all 5 are effectively represented within our clinical trial team, 4 of them as clinical trial sites. We also have Jim Bainbridge at Moorfields running the UK study, and between Moorfields and Bascom Palmer, you can pick 1A. Those are the 2 top eye hospitals in the world. So we've really managed to bring in some of the best knowledge leaders in this field who actually participate very closely with us. We have a meeting every October in which we bring all the surgeons together. Jim flies in from London, and the folks at Jules Stein fly out as well to sit down, go through the clinical data, and work through not only the design of phase 1, but now the design of what the phase 2 studies would look like as well. Procedure is really straightforward. It's actually done on an outpatient basis. It's done under, under sedation rather than anesthesia. It uses a standard off-the-shelf injection system that's a needle that tapers down to a soft tip cannula. As a vitrectomy followed by a PVD induction, and then a subretinal injection of the fluid that contains the cell. So once you've gone past the vitrectomy, the rest of the process takes all of about 90 seconds. As of today, we've treated 33 patients across the 3 enrolling trials. We have completely finished cohort 3 in both of the Stargardt's trials, the US and UK, as well as the US dry AMD study. We've treated 3 of the 4 patients in each of the 2 special cohorts that we have in the US. The third trial, which I'll talk about in a minute, is for myopic macular dystrophy. We are actually working with Jules Stein, hopefully looking to enroll the first patients in that study by the end of this quarter. In January 2012, we actually published a paper in The Lancet on the first 2 patients that were in the U.S. studies. It was one Stargardt's patient, one dry AMD patient, and what I can tell you is what we reported in that paper actually continues to be true across the boards in terms of the safety profile for the cells, and for many patients, the observations we made with respect to changes in visual acuity, improvements in visual acuity we've seen in many other patients in these studies. Those have been from visual acuity improvements that can be measured on an eye chart, but we also do VQF testing, which allows us to test things like reading speed, low light vision, and color perception. This just, again, from the paper, the first Stargardt's patient we treated when we started, she was hand motion in both eyes. She couldn't even count fingers. At 3 months, she was capable of actually not only counting fingers, but she was reading 5 letters on an eye chart, still hand motion in her fellow eye. She's now crossed through her 2-year follow-up and has maintained and actually had improvements in her visual acuity in her treated eye, and yet remains hand motion only in her fellow eye. She has been off immunosuppression for over, she's now gone through 2 years, 3 months. She's been off immunosuppression for over 2 years. So again, at this point we believe that these cells persist even in the absence of any immunosuppression, which was our goal. So going from Stargardt's and dry AMD, we also have been approved by the FDA for another disorder called myopic macular dystrophy. Again, like the other diseases, it's a disease that results in death of the RPE layer, followed ultimately by atrophy and death of the photoreceptors. In people with myopia, their eyeball is elongated. That causes the focal point to be ahead of the retina. For some of those patients, that elongation can actually create stress on their RPE layer, and the consequence is you get fissures that are created in the RPE layer, resulting in RPE atrophy, death, and then photoreceptor atrophy and death. In the US, MMD is the 7th leading cause of blindness. Because of the prevalence of myopia in Asia, in mainland China and Japan, it's the 2nd largest cause of blindness. Part of this is because it's an early adult onset disease, and as people live longer, you accumulate many patients who are ultimately blind from this disease. As I said earlier, the goal here is to start enrolling our first patients by the end of this quarter. Beyond what you can do with the native RPE cells, there are 200 macular degenerative diseases that we can attack altogether. We've also started looking at the ability, for second-generation products where we're using the RPE cells to deliver other agents which might have either cooperative or synergistic effects with the RPE cells that we're delivering, or can be used again to mitigate some of the damage that's already occurred, particularly in trying to prevent wet AMD from occurring in these patients. 90% of all wet AMD patients come through dry AMD. Our hope is that, by resurfacing the back of the eye with fresh RPE cells, they will in turn lay down fresh Brooks membrane, which will recreate that anti-angiogenic barrier. But we've looked at the ability to engineer these cells to express anti-angiogenic factors. There are various inflammatory components that have been implicated potentially in dry AMD. Complement factor D, C5, and C3 inhibitors have been tested in the clinics. They're all largely antibody-based, which means you can imagine, or receptor trap, that you can generate single-chain antibodies or receptor trap constructs that can be secreted by the transplanted RPE cells. Finally, the intellectual property platform is robust for this company. One of the kind of interesting features to being an embryonic stem cell company just birthed before George Bush took office was that when he came in and put a moratorium on all embryonic stem cell funding by the federal government, because we were not relying on federal money, it meant that we had no competition in this space. So it meant that being the first mover and first into the clinic has actually given us a really broad first-to-file patent system. So our original filings based on the derivation of these cells and the various pharmaceutical deployment of these cells, whether they be, again, in cell suspensions, the way we deliver them, whether you deploy them on a solid support or whether you just deliver them as a polarized layer, were all things that we thought to cover in these early filings. The language in our case was actually broader than embryonic stem cells, so it actually covers the type of pluripotent cells that iPS cells represent as well. Getting first into human patients meant we solved what I would call the final mile problem. And that is, we figured out some of the issues that were actually very significant to engraftment and functional performance of these cells. And so while sometimes these may seem like only incremental changes, from a commercialization standpoint, moving from, for instance, 25% viability to 95% viability is the difference between a commercial product and one that's not. And those are what are covered in some of our more recent filings. So I think we have a good lifecycle management strategy around our patent filings that comes with this as well. That leaves me 1 minute, so I will just end by saying, again, focusing on the eye, we're quite excited by the photoreceptor progenitor and ganglion progenitor programs that we have. In animal models for various forms of blindness, we've been able to demonstrate that the human photoreceptor progenitors when they're injected into the subretinal space as a suspension of cells, are actually able to migrate into the outer nucleated layer, differentiate the rest of the way into being rods and cones, and integrate it actually into the neurosensory retina in a way that you see functional enhancement of vision in those animals as well. And I will end there. If there's any questions, I'll take one or two now, and then I'll head out in the hallways so the next speaker can come up. Yes?
Two questions. When would you expect the AMD phase 2 results?
Yeah, I know Simon was going to ask that question as well. The end of phase 1 is still a ways off. What we have been, sorry, that's my timer. What we have been looking at is releasing through peer-reviewed publications interim data that we have available to us now. And so we've been working closely with all of the clinicians in the US, for example, on a co-authored manuscript that would provide a tremendous amount of detail around the safety studies, as well as some of the changes that we've seen with respect to visual acuity and from the VQF measurements as well.
So phase 2 might be what, 2015?
No, I think that, again, this is, there still is a long way to go between here and finalizing a phase 2 study for both the Stargardts and the dry AMD. Because the Stargardts is orphan, we actually think we could, in both cases, we think we could overlap them with the end of phase 1 and start phase 2 with the doses that we've already demonstrated are safe from phase 1, in part because, again, at this point, we have some patients with more than 2 years worth of data for them, and then amend the phase 2 protocol later. But it's all speculative at this point as to what the design will be. All I can tell you is, we've gotten a tremendous amount of input from not only the thought leaders, but we have consultants who've been in CBER, who've been in the MHRA, who are helping us work through the strategies, and then we're having direct discussions with both agencies about how to best streamline this process in a way that, in one case may produce a pivotal phase 2 study.
And then the second question was about patents.
Yes.
And just in general, what is patentable in these cells?
Well, I don't know if you follow what's been happening in Europe, but when the first embryonic stem cell patent got challenged, there was to a guy, Dr. Brustol, who was making neuronal cells from embryonic stem cells. It got knocked out on the basis that he had used a cell line in which the embryo had been destroyed. Since then, and actually as of last week, there's been a confirmatory decision from the European Patent Board of Appeals. They've reaffirmed that actually they will patent inventions where the invention was made using an embryonic stem cell that was not, that was made through a non-destructive technique, and they cite our papers as the example for making embryonic stem cells through a non-destructive technique. So I think from our perspective, we feel like we're on a pretty good trajectory, even in those jurisdictions where the underlying use of embryonic stem cells has had any impact on the patent system. But that being said, we've also far enough into the RPE portfolio that we've had good outcomes in the U.S. and other major jurisdictions. China, for example, we've got very broad patents in Australia, Japan.
And what specifically do you have in the U.S.?
So in the U.S., we have claims to methods of treatment using RP cells generated from embryonic stem cell sources. We have methods of manufacturing those cells, and then we're working through additional claims based on the pharmaceutical preparations. These cells, because they represent a cell that you ordinarily wouldn't get from adult tissue, actually have properties that make them distinct that gives you the ability to patent them as a composition of matter, particularly in the context of a pharmaceutical preparation. Gil's up next. I don't want to take any more of his time. So I'll be out in the foyer if anyone wants to chat any further.
probably on a monthly basis. So we're happy to get together when you stop by. Together.